This project is going to be covering all the bases of DNA and its role and all the factors it can affect. First, I'll talk about what DNA is. A DNA molecule is also known as deoxyribonucleic acid. It looks like two strands that twist around each other to form a double helix shape. It is also sometimes referred to as a twisted ladder form. It might just be the most important molecule there is. DNA has a special code that stores our genetic instructions. It is a code with 6 billion letters that contains the instructions of almost everything that lives. Every cell in a human body has 46 chromosomes, and each one of those also containing a DNA molecule. The chromosomes are squeezed tightly within proteins inside of the nucleus of the cell. Next, I'll talk about what DNA is made of. DNA is made up of tiny molecules which are called nucleotides. They are also given the name polynucleotide when formed in the DNA molecule. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. There are 4 different types of nitrogen bases, which are the following: adenine, thymine, guanine, and cytosine. The order of the nucleotides is very important. It is the DNA's instructions or genetic code for who we all are. Nucleotides are not simple and are very specific, as we are all made up of them. The sugar on the base also has another name, which is deoxyribose. The structure's backbone consists of sugars and phosphates bonded together. These backbones hold the DNA together and run down each side of the molecule. However, they are quite unique in direction and chemically run in different directions. These long chains are linked together by the nitrogen. At the top of one strand, the first phosphate connects to the sugar molecule's 5th carbon. This is called a 5 to the 3' chain. The other strand is vice versa. It begins with the sugar's 3rd carbon and the phosphate connect to the sugar's 5th carbon. The main part of the DNA model is the 4 bases. All of the bases are important in their own ways to a DNA model. Cytosine and thymine are linked together and called pyrimidine bases, while adenine and guanine are also linked together and called purine bases. These are the nitrogen bases that make up the 2 different kinds of nucleotide bases in DNA. First, I'll discuss adenine and thymine. Adenine and thymine bind through 2 hydrogen bonds. Their job is to assist in stabilizing. Thymine's job is to make directions and repair mutations. The 2 will always link together because of their 2 hydrogen bond structure. The other part of DNA bases are guanine and cytosine. Cytosine can be modified to make it very unique compared to the others. It is also for the study of gene mutation. Guanine's only job is to link up with cytosine. According to Chargaff's law, adenine will always bond with thymine because of the 2 hydrogen bonds. Same goes for cytosine, as it will always bond with guanine because of their 3 hydrogen bonds. This is why A and T will always be linked together and why G and C will always be paired. This also means that the A-T pair is much weaker than the G-C pair. For my final project, I also decided to build a DNA model of my own, which is playing on the screen right now. Each color is a different base, the red being adenine, yellow being thymine, blue being guanine, and green being cytosine. As you can see, the adenines are paired with the thymines, and the cytosines are with the guanines. The red and orange pom-pom balls represent the sugars. The red ones are also represent the weaker 2 carbon chain. The orange one represents the 3 carbon chain. The ribbon running down the side represents the phosphates. The order is all correct, including the Tata box, which is the weakest part of the DNA model. There are 4 roles DNA plays are replicated, encoding information, mutation/recombination, and gene expression. First, we'll talk about the replication, which is how it replicates and copies itself. Every cell in the human body has a copy of the same DNA. Due to this, it breaks the DNA strand in half and uses it as a guideline to building a new molecule so that it will have the same information. There's a huge part of DNA replication that includes a helicase. The helicase is important because it unwinds the DNA molecule, which breaks apart the bases and the hydrogen bonds. Now the DNA has a template to replicate. Then the strands become either the leading strand or lagging strand. The leading strand is where the replication starts. Then the bottom strand is called the lagging strand. The leading strand adds the matching bases through an enzyme called DNA polymerase. Before it matches the bases, the leading strand gets primed with the RNA primase, which starts the coding so the DNA can catch on. The DNA polymerase figures out the rest. The leading strand is easy to replicate because of its chemical order. So to properly replicate the lagging strand, it has to be primed by the RNA primase in small segments or sections. It goes through a whole process of getting primed by DNA polymers and RNA primers, then gets joined together by DNA ligase, which finalizes the replication by joining fragments. DNA polymers are also able to proofread over the DNA. This is because the replication process often mistakes the coding and will remove the nucleotides when they discover a mismatched base. Now let's take a look into mutation/recombination. A very important aspect to DNA is that it contains instructions for organisms to be able to develop, survive, and reproduce. To carry out these functions, the DNA sequences must be converted into messages that can be used to produce proteins. Mutations affect this area. Mutations happen through DNA chains as it is of genetic material. Since the bases A and T go together and G and C go together, an abruption in these links is how mutation happens. Things like chemicals, radiation may cause mutations. Also, mistakes in replication may cause a mutation. Mutations are often mistakes unless scientifically programmed to do so. There are two different kinds of mutations. First are gene mutations. DNA makes up genes which code for proteins. So when DNA has a mutation, there are different kinds of protein that are produced, which can affect an organism's trait. There are three different types of base mismatching. Substitution, which is when the base match up wrong with the wrong pair. Insertion, which an extra base is added in. And deletion, where a base is removed. And chromosome mutation, which has a few parts to it. Chromosomes are made up of DNA and protein and have a lot of genes in them. This chromosome mutation is where extra copies of that chromosome are generated and either mixed positioned with another, go into the wrong pair, get deleted, or replicated themselves. Next part of DNA is gene expression, which is also referred to as translation or transcription. This is where DNA is transcribed into RNA, which is then translated into proteins. Also keep that in mind, that RNA is DNA's cousins. This is very important as DNA written in code that needs to be made easier to read before a cell can make the proteins that are essential for life. RNA polymers works along the DNA chain to pull out certain bases and form a new chain with only half bases. The new piece is called out mRNA. There are two caps that get added onto the end of the RNA messenger to protect it from hazards and bad material. It is also to help it move out of the nucleus. The two caps are special of guanine and an enzyme with approximately 250 adenines. The next process of translation. Animal cells are also a big part in this process. Animal cells have ribosomes in which the messenger RNA gets fed into. Ribosomes are called rRNA and protein because of what they're made of. The rRNA purpose is to help the messenger RNA interact with the tRNA. tRNA translate the language of the mRNA into amino acid and protein language. This creates the polypeptide chain, which is the beginning of a protein. This entire process is complicated but important to understand as it is how DNA becomes RNA and then protein. DNA plays a huge part in evolution. As you remember from before, proteins have a role in creating the traits of an individual. If there are changes in the DNA sequence, this can change traits of an organism. Changes within molecules lead to microevolution of species. Small mutations may add up over time and change some of the functions of that organism. There are two circumstances that may or may not pass down mutations. If these mutations happen in normal adult body cells, then the changes do not affect future offspring. If the mutations happen in gametes or sex cells, those mutations do get passed down to the next generation and may affect the function of the offspring. These gamete mutations lead to microevolution. DNA is a very complex molecule that is the instructions for who we are. It is the main cause of evolutions and mutations. It is made of chromosomes and proteins to decide our traits. Overall, it is very interesting and complex and extremely important to understand. Thank you for watching my Biology 30 final.